We're going to start looking at today is how our muscles organize. So this is getting a lot into really how are these structured gross anatomy wise and how are they structured microscopic anatomy wise. I will say, especially with the microscopic anatomy, this feels like we're going overly deep. But as we get into how muscles go about contracting, we're going to see that a lot of the depth that we're going through in terms of the actual cellular structure ends up being important in how muscles actually contract. So we're going to see that, like a lot of the organs that we've talked about, there is a connective tissue component to the muscles. So we're going to see the muscles are surrounded by different layers of connective tissue. You have this epimysium, which surrounds the entire muscle, this paramysium, which is surrounding little bundles of muscle, and then the endomysium, which is surrounding each skeletal muscle fiber. And these help anchor this to the tendons and other parts of the skeleton. Do provide some space for nerves and blood vessels to travel through, and are going to provide some protection to that muscle. So if we look at a muscle here, you can see overall there is, and if you've ever had a steak or something else like that before, sometimes you might have seen that there's like this grizzle or silver skin that surrounds an entire muscle, especially if you're a hunter. This overall connective tissue layer that surrounds the outside of the muscle is known as this epimysium. It surrounds the entire outside of the muscle. It's made of dense irregular connective tissue. Then you can see within the muscle, we have these little bundles of muscle together, which a lot of times you might see on a steak or a pork chop. These are called fascicles. Each fascicle has a much thinner layer of dense irregular connective tissue surrounding it called a paramysium, or a round perimysium muscle. And then what we're going to have is areolar connective tissue surrounding each muscle cell or fiber, which that is called the endomysium. And again, these are all continuous within one another and are going to transition into that dense regular connective tissue tendon, which when overall this muscle can shorten, it will pull on the tendons, which will pull on the bones and bring about movement. And again, you can kind of see each of these pointing out where they would be on any particular part of this. So the endomysium, the epimysium, and everything else. And again, you can kind of see this right here. Here would be a fascicle. Here's the overall epimysium. Any connective tissue that would be in these areas would be paramysium, and then around each muscle cell or fiber, which are kind of used interchangeably because these are long continuous cells. A lot of times, like I said, referred to also as fiber. So if I say muscle fiber or muscle cell, it's really meaning the same thing. So the tendon, like I said, is at the end of these. This is what's going to connect these muscles to the bone. Again, dense regular connective tissue attaching muscle to bone or skin or something else like that. An aponeurosis is a flattened sheet of dense regular connective tissue. A lot of times you'd see these like with the abs, where you have a muscle that's pulling on a connective tissue structure but is not anchored to bone directly right there. So if you think about washboard abs, the raised areas would be the muscle. The little bumps in between those muscles are going to actually be those tendons or that aponeurosis. And you can kind of see that right here. This would be an aponeurosis, so would this one right here. And same with this one right here. These big bands of connective tissue. Again, lots of blood supply to muscles. They're very well vascularized. Again, especially since when you're working out, you want to deliver oxygen and nutrients and get rid of waste products. We also are going to have motor neurons that are traveling to these, again, coming off of the brain and spinal cord, depending on where the muscle's at. And it's going to be the end of these neurons called axons that are going to be coming into contact with each of these skeletal muscle fibers. And these axons are going to go through that connective tissue layer, and they're going to get in close contact, basically, or almost contact, with an actual muscle cell or fiber. And that connection, if this is the end of the axon right here, that synaptic end bulb, you're going to have it coming into close proximity, like when my two hands are here to one another with each other, and that is a neuromuscular junction. And this is going to be how the nervous system can actually stimulate that skeletal muscle and how we can control that through stimulus. So what we're going to look at now is, again, that muscle microanatomy. So what are these muscle cells looking like? So each skeletal muscle cell, like I said, is used interchangeably with fibers because they are quite long. As I was kind of saying before, they can be up to 30 centimeters long. Again, start out as these precursor cells called myoblasts that come together and fuse and become a multinucleated long cylindrical cell or fiber. And if we're looking at these ones, you can see this is what we're talking about. One of the things that's interesting with muscles is a lot of our prefixes and suffixes will change. So a lot of times we'll see myo, or the other big one is sarco. So I always kind of joke around, muscle cells are a lot like the Smurfs because Smurfs always say Smurf instead of another word. With muscle, we're always saying sarco instead of something else. So we're going to see the overall muscle cell membrane is called the sarcolemma. It is filled with that intracellular fluid called normally cytoplasm, but in this case it's sarcoplasm. The other thing that we're going to see, and this is going to be important to us a little bit later on, you have these invaginations or openings from the outside of the sarcolemma into the interior of the cell that are really kind of continuations of that sarcolemma called T-tubules because they cut across the cell in a transverse fashion. So transverse tubules are T-tubules. And you can see these can come in there and they surround each of these little myofibrils, which are surrounded by, so here's that T-tubule, and they come into close proximity with this blue area here, which is that sarcoplasmic reticulum. That is the smooth endoplasmic reticulum of a muscle cell. So again, sarcoplasmic reticulum. The other thing we see in muscle cells, lots of mitochondria. We're going to talk about this later on, but again, contraction requires energy in the form of ATP, and mitochondria are responsible for generating that. Lots of stored nutrients, things like glycogen in muscle cells as well, to provide the energy source for those mitochondria to convert it into ATP. So the sarcoplasmic reticulum is, like I said, the muscle's version of the smooth endoplasmic reticulum. It has a bunch of cellular protein pumps present in the wall, and these pumps are going to be moving calcium ions into that sarcoplasmic reticulum. So what we're going to see is there's little calcium pumps that can pump calcium into the sarcoplasmic reticulum, where they're kind of bound up by a couple different molecules called calmodulin and calcesquin. I can't speak it today. But this other one right here, it's sequestering it, so sequestrin, calsequestrin. There we go. Sorry, just looking at it and the word did not want to come out of my mouth. The other thing we're seeing here is voltage-gated channels that can open in response to a change in the cell membrane's voltage, and this would release that calcium out of the sarcoplasmic reticulum. This will be important to us later on. And again, the sarcoplasmic reticulum is surrounding each of these little myofibrils. These are a bunch of protein filaments called myofilaments or muscle filaments. And these are made up of what are called thick and thin filaments. And these are going to have proteins that make them up. So again, you can kind of see overall sarcolemma, invaginations of these T-tubules that are surrounding each of these little myofibril bundles with sarcoplasmic reticulum. But the membrane in reality is in contact or in close proximity to the sarcoplasmic reticulum, which are filled with calcium and are surrounding these myofibrils, which are made up of these thick and thin filaments. Like I said, these thick and thin filaments are what make up the myofibrils. And when we start talking about what are the thick myofilaments, these are made up of a protein called myosin. So a lot of times we refer to these thick filaments as myosin filaments or myosin fibers. A single protein of myosin looks like somebody was having a really, really bad day on the golf course and got so mad that they twisted their two clubs around one another. So you can see the tail here where it's spiraled around another, and then you have these myosin heads that look like the head of a golf club sticking out on the end. When we take, to get a myosin fiber, we tack a bunch of these together, as you can see right here, and you can have about 300 of these myosin fibers connected to one another to make this thick filament. Kind of looks like Jack and the Beanstalk, but you have all these myosin heads that are reaching out from this main, like this, from this main filament. The thin filaments are actually made up of 3 different types of protein. You have the main component, which is actin. Again, this actin is a bunch of these little beads that kind of go together here. This double-stranded bead, that is actin. And what we're going to see on actin, there is actually a site for those myosin heads to bind. So if this was the myosin fiber, the actin fiber, myosin heads should be able to reach up and connect to it, if that binding site is free. What we see normally is you have a couple of proteins surrounding this. You have this, around this actin helix, you have a protein called tropomyosin. When a muscle is not ready to actually do work or when it's at rest, this tropomyosin covers that myosin bonding site and blocks the actin and myosin from interacting from one another. We have a protein that's connected to this tropomyosin called troponin. And what we're going to see is a molecule combined to troponin and relieve that inhibition and let the actin and myosin interact. And we're going to see this is part of what happens with this contraction process. We're going to get the cell excited, and then you have what's called excitation contraction coupling, which is this process of freeing up the bonding sites on actin for the myosin so the actin and myosin can interact. And you can kind of see how these would be in proximity to one another. You have this actin filament right next to this myosin filament, this thick and thin filaments interacting with one another. And like I said, when the tropomyosin can get moved out of the way, the actin and myosin can interact, and that's when we can have muscle contraction. And these have a very ordered, at least in skeletal muscle, a very ordered repeating unit called a sarcomere. So you can see the purple in this image at the bottom. Those are those, and again, this is a 2D representation of a 3D structure. So again, we're looking at this in kind of like if the muscle fibers are under this, we're looking at it in kind of a side view. But you can see down here you have these myosin filaments, or these thick filaments that are in between these actin filaments, and you can see if we did this and then had a repeating unit much like this side right here over here, that is what we're looking at on the bottom of the screen is one sarcomere. We could have another one next to it on each side, and in reality we have thousands of these repeating units that make up these myofibrils. One of the things you'll notice on this one is you have areas where the thick and thin filaments overlap. Because this is such a structured, ordered fashion, those striations that we've been looking at on skeletal muscle and that, so that dark and light area, it is basically that overlap that we're talking about here, where the thick and thin filaments, where there's thick filaments, that is a region that's going to stain dark. And then the lighter region is where there's no thick filaments, so that is that alternating what we call A and I band, and that we'll get into that. But that is what gives skeletal muscle as well as cardiac muscle its striations, because these sarcomeres are very ordered in fashion. When we get to smooth muscle, it still has actin and myosin filaments, but the numbers change a little bit and their orientations change a little bit, so they don't give these visible lines that we can see. But definitely what we can see here is we have regions where there's only thin filaments, regions where there's only thick filaments, and then regions where they overlap. And again, these are these repeating units called sarcomeres. That is that basic unit of muscle, a sarcomere. If we kind of go back to this last picture here, this is what we refer to, because it's a zigzag line, that is that Z-disc on the end of each sarcomere. That is what is making up the end of a sarcomere. It's Z-disc to Z-disc, or Z-line to Z-line. There's proteins that help anchor these to one another so these filaments don't pull apart when stuff starts to contract. But again, the end of the sarcomere is those two Z-discs or Z-lines. Where we have overlapping filaments, that stains darker. Where the filaments overlap, they will stain darker. But where we have only thin filaments, that is the I band. So that is right next to the Z-disc. So that would be the light section. When you look at a striated muscle, that's the lighter area. The darker area is going to be what we call the A band, where you have the entire thick filament and the part that it overlaps, the thin filament. That would be the A band. Sorry, I went backwards. So if you're looking at an electron micrograph here, you can see dark, and we can see right here, dark, light, A band, I band, A band, I band, and that repeating over and over again is what gives these muscle cells or fibers their striated appearance. With a lot of these ones, I do want to tell you with these letters and stuff like that, I don't get real crazy on these. I want you to know I band and A band, and that Z lines are the end of this, are kind of what delineates or defines what a sarcomere is. There is some of these other areas that they do name on this, and it starts to me, in some cases, be a little bit much, but the H zone is that central portion of the A band where there's only thick filaments present, When the muscle is fully contracted, this completely disappears. The M line is that connection point where those thick filaments back up to one another. Again, if you see these terms, that's why I'm defining these. To me, I'm not going to worry about like the H zone and the M line. Like I said, no A band and I band, know what the Z lines are. But again, it's this structured pattern over and over again that is going to give you those light and dark regions in the microscope, that striated look. So you can kind of see it here now. Here would be one sarcomere right from here to here, another sarcomere from there to there, another sarcomere from there to there. So you can see each part of these I bands, I band, A band, I band. So the I band is kind of the end of one sarcomere and the beginning of the next. The A band is that area where those thick filaments are. And again, this is in 3 dimensions, so you can kind of see the whole layout here, a little bit more zoomed in, and some of what's going on with that. Again, to me, knowing the Z discs and the sarcomere, knowing the A band and the I band are what give you those striations. To me, those are the key things that I'm worried about. The M line and the H zone, I'm probably not going to ask you about. The other thing to kind of realize is the orientation of these things. If you start seeing where the thick and thin filaments interact with one another, you're going to have one myosin filament surrounded by and interacting with, in a lot of cases, 6 different thin filaments, because those myosin heads go out in all directions. So what I was trying to show with this one is, again, we've been looking at a 2D representation of that where these things are overlapping one another. Realize this is happening in 3 dimensions in a cylinder. So just keep that in mind. So there's a lot of interaction between these actin and myosin filaments. There's a few other functional proteins. You have something called Connectin going from the Z disc to the M line, which is helping be the core of that thick filament, helps hold those thick filaments in place. Spring-like tension during contraction on this one. You have something called nebulin, part of the I band, keeping that structure together a little bit. Something called dystrophin that anchors the myofibrils to the sarcolemma on each end. If you have something called muscular dystrophy, this protein is abnormal and doesn't function correctly. But again, if we now look at a zoomed in slide, a light micrograph of this, you can see the A band and the I bands as light and dark regions that would make up this skeletal muscle. And again, lots of T-tubules going down there, those invaginations of that sarcolemma. This is going to be important as we get to contraction, because there's going to be changes in this membrane that need to be taken to the interior of the cell for all this interaction and this contraction to work. So we will get into all that, but a lot of this was just trying to explain all the parts. So when I start going through them in the contraction process, you have some understanding of what is going on in each of these parts. So in reality, if we start looking at this one, if we were looking at a sarcomere down on the bottom here, you can kind of see if this was a Z line and this is a Z line, you have these thick filaments pulling the thin filaments closer together. Overall, that shortens the sarcomere. So if we were to do it with my elbows, if these were 2 filaments and here's the ends, each of the elbows, if you start pulling the centers together, you're going to see the ends of the elbows get closer together. That is what's happening when a contraction occurs. These thick filaments pull the thin filaments past them. It moves those Z lines closer together because we are shortening the amount of space in that I band right here and making it disappear as we contract it. That overall, because we're doing this thousands of times over the length of the whole muscle, as you do that, that muscle is going to get shorter, pulls on the tendon, it brings about movement. So this is referred to as the sliding filament theory. But again, you can see if you pull these thin filaments, each end here, if you pull these towards the center, the distance from that Z disc to that Z disc is going to get shorter. And like I said, we have thousands of these backed up to one another. If you do that 1,000 times over, that whole fiber is going to get shorter. And you do that with a bunch of fibers, a bunch of muscle cells, the muscle will get shorter, and you bring about contraction. So that is kind of the basics of the anatomy on this one. What we're going to get into on the next one here is a lot of, to me, the deepest physiology, where we're going to be getting into how do we stimulate the muscle and how does that lead to contraction. So I will talk to you again next time.